turn on genes, so we have transcription as a mechanism. We mentioned in the context of the alkanitase moonlighting enzyme, this notion that messenger RNA degradation can control how much protein is made, just by how long a messenger RNA can hang around. There's also direct ways to control the rate of a protein synthesis on the ribosome. And then once we've got the enzyme that we're looking at, it can, of course, be degraded by either targeted pathways or by sort of non-specific chemical events that occur to it. And then we have an array of mechanisms that are more on the lines of the kinds of things that we talk about in this class. Sequestering enzymes in certain organelles. Binding substrate, well, how much substrate is around. Right? Making product, but can the product come back and inhibit it, perhaps, if there's a lot of it? Where are we on the V versus S curve? Can we move on that? What does the V versus S curve even look like? Is it a Michaelis-Menten hyperbolic plot, or is it an allosteric sigmoidal plot? And what does that mean? Where is Km with respect to how much substrate there is in the cell? Allosteric effectors, covalent modifications, combinations with other molecules like other proteins or RNAs. Right? All mechanisms that we can use to control both the number of molecules and how active each molecule is. Okay. So here's a picture of what's called a metabolome. And at any given state of a cell, let's say E. coli this is E. coli growing on glucose, so that means in its happy growth phase, where it's doubling as quickly as every 20 minutes. It's doubling the number of cells. You go in there and grab some cytoplasm. E. coli is only one compartment, and ask, well, what kinds of small molecules are in there? So the metabolome doesn't refer to the protein so much. That would be the proteome. The metabolome is what are all the metabolic molecules that are present in vivo, right? And so this is just a snapshot of an E. coli growing on glucose. It's remarkable what fraction of the total molecules are one molecule, the same as glutamate. And as we'll see later, glutamate is a central molecule that bridges carbon and nitrogen metabolism and also feeds a lot of other amino acid metabolism. So it kind of makes sense that there would be a lot of glutamate. I was pretty shocked when I saw how much more glutamate, though. Nucleotides, carbon, sugar molecules, etc. And mostly you have things like here, other amino acids. So in this tiny little segment, you've got many other amino acids, not just the coding ones, but all the ones that are things like homocysteine, for example, ornithine. Key amino acids form metabolic pathways that are just don't happen to be in the genetic code. So how do you measure this? Mostly by mass spectrometry, if you're looking at the metabolome. If you want to look at the proteome, like which proteins are around at any given time for a cell that's differentiating, you can run 2D protein gels and then identify the proteins in those gels, typically by mass spec. Or if you want to look at just the nucleic acid levels, you can do DNA microarrays and with all the genes in an organism, and then isolate your bulk messenger RNA and do a hybridization. And then if you have a printout of your chip, it tells you which genes are which location on a chip. Some of them will light up because there'll be a fluorescent tag on there, and then it'll tell you, well, which of your mRNAs are expressed at that time and which are not. So you change the regulatory state of the cell, you give it more food or something else happens, and then a large pattern of messenger RNAs may shift. Right? So we're not that clued in as to all the mechanisms that would make that happen, but we can at least see what the change is and then think about it. Regulation at the level of individual enzymes. This is really a reminder, but one aspect of this is to say that there's two aspects to this. One is that the Km of the enzymes has some tendency to match the metabolite concentration. This is a very loose term. So here's the Km of enzymes. Each of these points would represent an enzyme that metabolizes these different kinds of metabolites. So other is mostly what you see, but there's NAD specifically, ATP, NADPH. And so here, for example, the Km of the enzyme is 10 to the minus 4 molar. The metabolite concentration is 10 to the minus 4 molar. You would be on this black line here. And so what this is trying to say is that most of the enzymes are between here and here. Some are outside, but many of them are between here. So you're within tenfold. That is to say, the concentration of the metabolite in the cell is within tenfold of the Km of the enzyme. And for the ones that are not, there's a lot more where the metabolite concentration is a lot higher than the Km. That's signaled by the fact that they're way up here, and many fewer that are down here. So that's kind of one general principle of regulation that we can look at, is to think about where we're at, because the level of the metabolite in the cell that the enzyme is binding tells you where on the V versus S curve you are. Right? Are you up at Vmax, or are you down, way lower? Right? Because your Km is much higher than your substrate metabolite concentration. Now, this is telling you that that doesn't happen very much, because there's not very many enzymes that are down here. And then the other part of this is, what does the V versus S curve even look like? We talked about the Michaelis-Menten kind of plot where there's no cooperativity. That would correspond here to a Hill coefficient. That would be 1. So the required change in the substrate concentration to increase velocity from 10% of Vmax to 90% of Vmax. Right? So you're pretty much covering your whole V versus S plot there, from the 10% level to the 90% level. So how much more substrate do you need? So if you have no cooperativity, that means your plot is not sigmoidal at all, then it takes you 80 times more substrate to be at 90% as opposed to 10%. So it might be very difficult for the cell in its regulatory processes to come up with that much more if it really wanted to speed up an enzyme by so much, from 10 to 90 on the Vmax curve. But if you're cooperative, and this basically tells you how much cooperativity there is to a maximum of 4. And this is similar to what it means in hemoglobin. Then the required change in substrate concentration is much less. And you can already see, even for a relatively mild cooperativity, you've got a huge difference, only 9-fold is required instead of 80-fold. Okay, so that's an important aspect of regulation as well, and the regulated enzymes, as we've already talked about, are the ones with the sigmoidal curves, where smaller changes in substrate concentrations can have larger effects on activity. Right. Any questions? Okay, so you've seen this slide already. So we have our delta G is delta G0 prime plus RT-LNQ. And that is our key equation with respect to understanding how regulation works. So here's the standard state conditions. This is a review. So glucose, this is the mutase that we've already talked about today. Converting these two metabolites. K prime equals to standard state is to the right. The delta G0 prime is minus 7.3 kilojoule per mole. If you start the standard state of 1 molar of both products, both substrate and product, or both metabolites, the positive equilibrium constant and negative delta G tell you that you're going to move to the right. And the magnitude of the thermodynamic driving force has to do with where you start as compared to where you finish. So if you start everything at 1 molar, and you finish at 19, the driving force is minus 7.3, right? But if we're in the cell and we have different concentrations, right? So we have, say, 20 millimolar of this and only 0.1 millimolar of that. Right? Now we've got 200-fold more of this than this, but equilibrium is 19. So the driving force will move you backwards, and the delta